Enzyme kinetics, the study of enzyme catalyzed reactions. Enzymes are essential proteins that catalyze chemical reactions in living organisms. They play a crucial role in facilitating biochemical processes, allowing organisms to metabolize nutrients, synthesize new molecules, and maintain homeostasis. Enzyme kinetics is the study of the rates and mechanisms of enzyme-catalyzed reactions. Understanding enzyme kinetics can shed light on the behavior of enzymes and provide insights into their function and regulation. In this article, we will explore the principles of enzyme kinetics and its applications in various fields of biology and medicine. Enzyme kinetics basics. Enzyme kinetics involves measuring the rate of a reaction catalyzed by an enzyme in the presence of different concentrations of substrates, inhibitors, and activators. Enzyme kinetics provides a quantitative framework for understanding the effects of these factors on enzyme activity and for characterizing the properties of enzymes. The Michaelis-Menten model. One of the central concepts in enzyme kinetics is the Michaelis-Menten model, which describes the relationship between the rate of an enzyme-catalyzed reaction and the concentration of substrate. The Michaelis-Menten model is based on the following assumptions. The enzyme-substrate complex is formed reversibly. The rate-limiting step in the reaction is the conversion of the enzyme-substrate complex to the product. The rate of the reaction is proportional to the concentration of the enzyme-substrate complex. The Michaelis-Menten equation is a mathematical formulation of the Michaelis-Menten model and is given by V equals V max S Km plus S, where V is the rate of the reaction, S is the concentration of substrate, V max is the maximum velocity of the reaction, and Km is the Michaelis constant. The Michaelis constant is a measure of the affinity of the enzyme for its substrate and is defined as the substrate concentration at which the reaction rate is half of the maximum velocity. The Michaelis-Menten model has several important implications. First, it predicts that the rate of the reaction will increase linearly with substrate concentration at low concentrations, reaching a maximum value at high concentrations. Second, it shows that the rate of the reaction depends on the relative values of the kinetic constants, Vmax and Km, and the concentration of substrate. Therefore, the Michaelis-Menten equation can be used to estimate these constants from experimental data. Enzyme inhibition. Enzyme inhibition is the process by which a molecule binds to an enzyme and reduces its activity. Enzyme inhibitors are essential tools for studying enzyme function and for developing drugs that target specific enzymes. There are two types of enzyme inhibitors: competitive inhibitors and non-competitive inhibitors. Competitive inhibitors bind to the active site of an enzyme and compete with the substrate for binding. They can be overcome by increasing the concentration of substrate. The effect of a competitive inhibitor on enzyme activity can be described by the following equation. V equals V max S Km 1 plus I/K plus S, where I is the concentration of inhibitor and K is the inhibition constant. Non-competitive inhibitors bind to an allosteric site on an enzyme and do not compete with the substrate for binding. They reduce the activity of the enzyme by altering its conformation or stability. The effect of a non-competitive inhibitor on enzyme activity can be described by the following equation. V equals V max 1 plus I/K S Km plus S, where I is the concentration of inhibitor and K is the dissociation constant. Enzyme inhibition can have important physiological and pathological consequences. For example, many drugs act as enzyme inhibitors and are used to treat diseases such as cancer, hypertension, and HIV. Understanding how enzyme inhibitors interact with their targets is essential for the rational design and optimization of new drugs. Enzyme activation. Enzyme activation is the process by which a molecule binds to an enzyme and increases its activity. Enzyme activators are less commonly used than inhibitors in research and drug development, but they play important roles in metabolism and signaling pathways. The mechanism of enzyme activation varies depending on the enzyme and the activator. Some activators bind to the active site and enhance substrate binding, while others bind to allosteric sites and induce a conformational change that increases enzymatic activity. The effect of an activator on enzyme activity can be described by the following equation. V equals Vmax, kmt. Kmt plus, S, 1 plus, A/Ka. Where kmt is the Michaelis constant in the presence of the activator, A is the concentration of activator, and Ka is the activation constant. Enzyme activation is an important mechanism for regulating metabolic pathways and ensuring that enzymes are active only when needed. For example, the activation of enzymes involved in glucose metabolism by insulin is critical for maintaining blood glucose levels within a narrow range. Applications of enzyme kinetics. Enzyme kinetics has a wide range of applications in biology, medicine, and biotechnology. Here are some examples. Drug discovery. Enzyme inhibitors and activators are important targets for drug discovery. By characterizing the kinetic properties of enzymes and their interactions with inhibitors and activators, researchers can identify new drug leads and optimize their potency and selectivity. Metabolic engineering. Enzyme kinetics is a valuable tool for designing and optimizing metabolic pathways in microorganisms and plants. By manipulating the kinetic properties of enzymes and substrates, researchers can increase the yield of desired products and improve the efficiency of bioconversion processes. Diagnostic assays. Enzyme kinetics is the basis of many diagnostic assays used in clinical and research settings. For example, enzyme-linked immunosorbent assays, ELISAs, use enzyme catalyzed reactions to detect the presence of antibodies or antigens in biological samples. Enzyme engineering. Enzyme kinetics is used to design and optimize enzymes with specific properties for industrial and biotechnological applications. By introducing mutations into enzymes and screening their kinetic properties, researchers can identify enzymes with improved activity, stability, and selectivity. Enzyme kinetics is also useful in the study of enzyme inhibition and activation. Enzyme inhibition is the process by which a molecule binds to an enzyme and reduces its activity, and it can have important physiological and pathological consequences. For example, many diseases are caused by enzyme abnormalities, and understanding how enzyme inhibitors interact with their targets is essential for developing effective treatments. Enzyme activation, on the other hand, is the process by which a molecule binds to an enzyme and increases its activity. Enzyme kinetics has a wide range of applications in biology, medicine, and biotechnology. For example, drug discovery is an area where enzyme kinetics is crucial. Another area where enzyme kinetics is useful is metabolic engineering. By manipulating the kinetic properties of enzymes and substrates, researchers can increase the yield of desired products and improve the efficiency of bioconversion processes. Enzyme kinetics is also applied in diagnostic assays, where enzyme catalyzed reactions are used to detect the presence of antibodies or antigens in biological samples. Enzyme kinetics is an important area of study, but it is not without limitations. Additionally, the model assumes that the rate-limiting step is the conversion of the enzyme substrate complex to the product, which may not always be true. To overcome these limitations, more complex models have been developed, such as the Briggs-Haldane model and the Hill equation. These models take into account additional factors that can affect enzyme activity, such as cooperativity and nonlinear kinetics. In addition to its applications in biology and medicine, enzyme kinetics is also an important tool in industrial biotechnology. Enzymes are widely used in industries such as food and beverage, detergents, and pulp and paper, and their kinetic properties are crucial for designing and optimizing these processes. By understanding the kinetic properties of enzymes and their interactions with substrates, inhibitors, and activators, industrial researchers can develop more efficient and cost-effective processes.